The first drug manufactured using recombinant DNA technology was insulin, which is produced in bacterial cell E. coli. Before 1982, people with type 1 diabetes mellitus obtained insulin that they injected daily from pancreas removed from cattle in slaughterhouse. Cattle insulin is so similar to the human peptide, differing in only two of its 51 amino acids that most people with diabetes could use it. However, about 1 in 20 patients is allergic to cow insulin because of the slight chemical difference. Before recombinant DNA technology, the allergic patient had to use expensive combination of insulin from other animals or from human cadavers. Insulin is a peptide and is therefore straightforward to mass produce in bacteria. Some drugs, however, require that sugars be attached to the peptide or protein, or that the protein fold in specific intricate ways to function. Transgenic non-human mammals can express human genes in their milk, including genes that encode clotting factors, clot busters, and connective tissue protein collagen. Human antibodies produced in rabbit and cow milk, for example, are used to treat cancers. Malaria remains one of the African deadliest diseases, killing nearly 500,000 children under the age of 5 and accounting for approximately 95% of global malaria cases and 96% of the deaths in 2021. Nearly every minute, a child under 5 years old dies of malaria. A vaccine that protects against malaria is based on altering a bacterium that normally inhabit the mosquito gut. Mosquito transmit Plasmodium falciparum, the parasites that cause malaria. The bacteria are given gene from E. coli bacteria that enable them to produce protein that tear apart of the insect intestine. Using recombinant bacteria is easier than genetically modified mosquito to prevent malaria. Atlantic salmon that grow faster on less food than unaltered salmon due to genes from Chinook salmon and a gene from eel-like ocean food is an example of transgenic organisms. The vaccine for COVID-19, messenger RNA vaccine, is made using messenger RNA that gives your cells instruction for how to make the spike protein found on the surface of the COVID-19 virus. After vaccination, your immune cells begin making the spike protein and displaying them on the cell surface. This causes your body to create antibodies that can fight the COVID-19 virus. Viral vector vaccine is made when genetic material from COVID-19 virus inserted into the unrelated harmless virus. When the viral vector gene gets into your cells, it devours genetic material from COVID-19 virus that gives your cells instruction for how to make the spike protein found on the surface of the COVID-19 virus. Once your cells displace the spike protein on their surface, your immune system creates antibodies that can fight the COVID-19 virus. Subunit vaccine include only the parts of the virus that best stimulate your immune system. This type of COVID-19 vaccine contains harmless S proteins. Once your immune system recognizes the S protein, it creates antibodies, and defensive white blood cells. If you later become infected with COVID-19, the antibodies will fight the virus.